Your red blood cells carry oxygen to every tissue in your body. And they cannot use a single molecule of it themselves. That's not a limitation or a design flaw. It's deliberate, it's constructed on purpose, and the way they achieve it is one of the stranger things I've come across. Let me start with the basic fact. A mature red blood cell has no mitochondria. None at all. Think about what that means. Every other cell in your body uses oxygen to run its electron transport chain and produce ATP. That's the entire point of breathing. Red blood cells can't do any of it. They have no chain to run. So how do they power themselves? Glycolysis, the breakdown of glucose in the main body of the cell, without oxygen. It's the older pathway, the one cells used before oxygen was available in the atmosphere, and it produces a small fraction of the energy per molecule of sugar compared with the full oxygen-dependent route. That's all a red blood cell gets. And it's enough, because a red blood cell isn't doing much. It's a container. It holds hemoglobin, it changes shape to squeeze through capillaries, and it maintains itself. Those jobs are cheap. Alright. Now the interesting part. Red blood cells are not born without mitochondria. Earlier in their development, in the bone marrow, precursor cells have mitochondria like anything else. They're normal cells doing normal cellular things. And then, at the final stage of maturation, they eliminate the entire cohort. Every mitochondrion, removed. I want to be precise about that word. This is not the mitochondria wearing out or being lost. There's a dedicated program that identifies them and clears them, deliberately, as part of becoming a red blood cell. They do the same thing to their nucleus. It gets pushed out entirely. A mature red blood cell has no DNA. It's the only cell type in your body that discards its own genome as a developmental step. So the cell strips itself down. Nucleus out. Mitochondria out. What's left is essentially a bag of hemoglobin with a flexible membrane. Now here's the sequence I find genuinely elegant, and it connects to a video I made last Thursday. Hemoglobin is built around heme. Heme is what actually binds the oxygen, and it's what makes blood red. And heme is manufactured inside mitochondria. So the developing red blood cell needs its mitochondria in order to build the very cargo it's going to spend its life carrying. It uses them to produce heme, incorporates that heme into hemoglobin, fills itself with the finished product, and then it destroys the machinery that made it. Build the cargo hold using the engine, then throw the engine overboard. The mitochondria are essential right up until the moment they're not, and then they're gone. How does that demolition work? There's a protein called NIX, sometimes written as BNIP3L. It sits on the outer mitochondrial membrane and it gets switched on specifically during the final stage of red blood cell differentiation. NIX acts as a receptor. It marks mitochondria for engulfment by autophagosomes, which then deliver them to lysosomes for destruction. In animals lacking NIX, the reticulocytes have a significant defect in clearance. The mitochondria don't get removed properly. And here's a detail I particularly like, because it connects to yesterday's video. Yesterday I talked about cytochrome C escaping from mitochondria and triggering the cell death cascade. You might assume this clearance uses the same machinery. Kill the mitochondria with the death program. It doesn't. Researchers tested that specifically. Mitochondrial clearance in reticulocytes doesn't require the standard proptotic proteins. NIX isn't working through the death pathway, which makes sense. The cell isn't dying. It's maturing. You want the mitochondria gone and the cell intact, and using the demolition system designed to take the whole cell apart would be exactly the wrong tool. So evolution built a separate one. Same target, opposite intent. Now, why do this at all? Two reasons are usually given, and both are practical. The first is space. A red blood cell's job is to carry as much hemoglobin as possible. Every organelle inside it is volume not spent on cargo. Removing the nucleus and the mitochondria frees up a large amount of room. The second is more pointed. Mitochondria consume oxygen. That's what they do. A red blood cell full of working mitochondria would be metabolizing some of the very oxygen it's supposed to be delivering elsewhere. A courier that eats the parcel is a bad courier. So the courier is built without a mouth. There's a third consideration too. Red blood cells spend their lives handling oxygen and iron together, which is a combination that generates reactive oxygen species readily. Fewer membranes and no mitochondrial electron transport chain means fewer places for that chemistry to cause trouble. In a cell that has no nucleus and therefore cannot manufacture replacement proteins. That last point is worth pausing on. A red blood cell can't repair itself in any meaningful way. No DNA means no new protein synthesis. Whatever it's built with is what it has for its entire lifespan, which is around 4 months. So minimizing internal sources of damage isn't optional. Now let me be honest about the state of the science. The specific molecular details of how this clearance happens are described in the literature as still controversial. NIX is clearly involved, and so is another regulator called OLK1. But the full picture, exactly how the mitochondria are selected and processed, is not settled. So I'm giving you a solid finding with a contested mechanism, and I'd rather label that than present it as tidier than it is. What's the takeaway? There's no lever here. Nothing to do, nothing to change. This is developmental biology, and it's already finished happening. What I'd offer is the reframe. We talk about mitochondria as though more is always better and every cell wants as many as it can get. But your body deliberately builds a cell type with none, uses a dedicated program to make sure of it, and that cell type is one of the most numerous you have. Sometimes the right number of mitochondria for a job is 0. The cell that carries your oxygen is the clearest example there is. Now, where my recommendation sits, and today it's genuinely outside the topic. CoQ10 is a carrier in the electron transport chain. Red blood cells don't have one, so nothing in this video applies to them at all. It applies to essentially every other cell in your body. Levels tend to decline as we age. The link in the description below goes to a free 5-item morning checklist. Mitolin's on that page as well, and it's an affiliate link. I earn a commission at no extra cost to you. And if you want more of this, subscribe. This video is educational only and it's not medical advice.